My name is Beth Leigh. I'm an associate professor of microbiology here at the Department of Biology and Wildlife and in the Institute of Arctic Biology at UAF. I certainly collaborate with other faculty within the Department of Biology and Wildlife. It's a very diverse department. We have everything from moose biologists to microbiologists. I think we have to really be conscious and make a conscious effort to become broadly educated and maintain the ability to communicate across disciplines if we're going to take on the sorts of grand challenges that we're faced with in the 21st century. We have to understand how the natural world works. So how do plants function? How do they interact with microorganisms? How do microbes break down chemicals in the environment? What I came to learn through the course of my master's research is that plant roots really stimulate microorganisms in soil to biodegrade contaminants. And we can take that information and apply it to do something like clean up a contaminated site by planting the appropriate plants there that would then nurture the appropriate microorganisms to clean up that soil. And it's really meaningful to me to feel like what I'm doing is having an impact here. I work closely with the state, the Alaska Department of Environmental Conservation, and when they have a new contamination issue, they often call me and ask me if I might be interested in studying it, and they often fund students in my lab to do experiments on those sites, and we're even implementing some of our cleanup technologies now in some rural Alaskan environments that are really suffering from contamination issues. And so often students will reach out to me and we'll talk through what their career goals and interests are and what kinds of skills they might want to develop and try to find a project that would match those skills. So just last week we were up in Barrow. I was up there with a Ph.D. student collecting seawater, and we've filled a cold room now with 286 bottles of Arctic seawater with crude oil in them, and we're monitoring the biodegradation and examining the microbial response to oil spills to better understand if there was an oil spill in the Arctic, how long it would last, if it would biodegrade, and also how to respond to that oil spill in a way that would maximize biodegradation. In microbial ecology, one of the limiting factors in studying microorganisms is our ability to detect them. So historically, we only had initially microscopes, so we could only visualize them. DNA sequencing technologies have advanced so rapidly that now we're in a really exciting stage. Here at UAF, we're lucky to have the DNA Core Lab, where we have some of the newest sequencers on the market, and even one that's not even on the market just yet. We're part of an experimental pilot program to test it out, and we're using it now to sequence entire communities of microorganisms and all of their DNA from thawing permafrost and from Alaskan Arctic environments. It's one of the fun things about our line of research is we have this combination of field and laboratory research. And so there are these great opportunities to go out to Arctic and subarctic environments really readily to collect samples and then bring those back in the lab to do their studies. So we've done fieldwork everywhere from the Arctic Ocean to the boreal forest and everything in between.